Alô, alô. Alô.
Alô?
That's all good. I'm just going to go make a coffee quickly.
Now I'm looking at you, Renato.
In YouTube?
In YouTube.
Wow.
Wow, marvelous, maravilloso. Thank you so much. And you too, as well. Gabriel Fernandez. Hi. ¿Cómo están vocês?
Você sabe como é que é, né? Até agendar. Mercedes, em 5 minutos começamos, ok?
Ah, perfeito.
Hi everyone, it's a pleasure to stay here in the first Devonian Gondwana Symposium, and bem, falarei em português também, mas é muito importante, it's very important, participation of the scientists. Devonian it's a moment very, very awesome for me. And we will start the day of works with the talk de Elizabeth Dowding, Devonian Biogeography and Global Correlation, Reclaiming Gondwana. We will see this talk in YouTube, and the questions about the talk nós faremos após a palestra, ok? Muito obrigado, então. Sejam bem-vindos e iniciemos os trabalhos. Let's start.
Hello, everyone. My name is Elizabeth May Dowding, and I'm currently sitting at the University of Oslo at the Center for Earth, Environment and Dynamics. Before I start my talk, I would just like to acknowledge the Gadigal people of the Eora Nation, which is where I currently am in beautiful Australia, down on the East Coast. I'm really grateful for the opportunity to be able to speak to you all today, and I really look forward to discussing some of what really excites me about Devonian biogeography, but also some of the problems that I'm seeing. I am primarily a Devonian biogeographer, and I work with the Homolonoideae, which are a group of Phacopa trilobites. So we'll be talking a lot about global correlations and the reclamation of Gondwana, but I'll also try and sneak as much of those trilobites in as I possibly can. I would absolutely love to thank the organizers of this, and I am incredibly grateful to be the first speaker of the first Devonian Gondwana Symposium. It's absolutely fantastic initiative, and I'm beyond pleased to be here and to be with you all. So yeah, thank you very much, and I guess we'll get into it. So to summarize my talk today, I'll be talking a lot about the way that we've studied and the way that we've considered Devonian Gondwana correlations, and how that is often biased by a Eurocentric focus. I've spoken to my colleagues about this quite a few times, and I know that the notion of how our perspectives and conditions of research can change and have changed our interpretations of data can kind of put some people off. There's this idea within science that there's true objectivity that's attainable. But I really believe that we need to be aware of our positionality. We need to be aware of how we engage with science and engage with data, and how we communicate that to other people who are outside of our expertise or even outside of our local areas. And so I think that throughout this talk, I'll be really advocating objectivity as kind of a goal or an ideal rather than a true feature. And so talking about sampling bias, observer bias, and how that's interrelated with historicity and the legacy of post-colonial societies, for example, Australia, how that has really changed not just our modern lives as people in those countries, but also our interpretation of data and the patterns that we see even within the Devonian. So yes, I will be focusing mainly on studying and considering Devonian Gondwana correlations and how that is biased by a Eurocentric focus and Eurocentric correlations as a starting point. And to move through that talk, I will then end with the ecological and evolutionary patterns brought in by my favorite group, the Homolonoideae. Okay, so I guess we can get into it. So first things first, we have our foundations. And I'm sure that everybody here knows it, given that we are Gondwanan scientists. But for some frame, the Devonian was 419 million years to 358 million years ago. And during this period, Gondwana was 70% of the continental mass. Not sure where that second 7 came from, but and its modern geopolitical areas covered include my own beautiful country, Australia, and many countries here in the global south. So as part of this framing of my talk, I think it's really important that I differentiate between Gondwanan areas and the global south, just so that we don't get confused. So when I'm talking about the global south, I will definitely be talking primarily about modern geopolitical areas and the histories that go with that in terms of human histories and the way that we engage with science. And if I'm talking about Gondwanan areas, then I am speaking in a more biogeographical sense. I will be talking about my fossil data, and I will be talking about environments and things. My main feature in all of my research is bioregionalization, which is a subset of biogeography. And I just want to differentiate that from those modern geopolitical areas. So Gondwana was really massive, as we've probably got from the 70%. And the Devonian is a really significant piece of time. And the taxic evolutionary developments and marine communities in this area were really diverse, and the tectonics were really dynamic. And so when we're looking to study and correlate between all of these and really uncover some scientifically interesting features, it can be quite easy to be overwhelmed or bamboozled by all the different ways that the data can lead you into something new and exciting. And I know for myself, I've spent so many years getting excited about all of the things that are happening in the Devonian and how I can draw them all together to create this kind of consistent, cogent narrative and explanation of what I think was happening during the Devonian in terms of biotic and abiotic interactions. And so because of that, I decided pretty early on to work in biogeography. Because I'm selfish and I don't want to have to pick one, thought biogeography was the perfect place for me. I could pick all of them and study them all at once. So biogeography is a multidisciplinary field that seeks to disentangle and really get into those dependent relationships of life on Earth. And I love studying biogeography because I can ask questions at scale, and I can see how patterns change within the data based on that scale. And so I'm really conscious of how the way that we're viewing the data, the way that we're manipulating the data, the scale that we're looking at the data on really does change the pattern and therefore the interpretation. And dealing with biogeography, I also get to pull data that other people have made. I can create data myself. For example, I've created my own databases on Australian Devonian fauna. And I can also ask for really cool collaborations with geophysicists and geochemists and stratigraphers and taphonomists to do something fun and new. And it's honestly really hard to not get excited about all the opportunities to study the Devonian. So my own group are trilobites, and so I have these beautiful true tops here. And that's not my group. I just thought the picture looked really nice. But my group are the hominidae I've mentioned, and they give me the evolutionary and taxonomic foundation for the work. And I think that for people who deal with data and who are looking at these global correlations, having something to really get your teeth into and to underpin all of these broad scale things that we're talking about is super important, because it means that you're not getting lost in the data, and it's easier for you to pick out what patterns are likely based on your understanding of your group. So now we begin properly. So this is an image from my 2020 paper where I looked at the accepted Devonian bioregionalization. So the bioregionalization that is currently being used, you would have heard about, and it features the Old World realm and the Malvino-Caffric and the Eastern Americas realm. And it all kind of started with Art Bucco et al. In 1969, and they created a zoogeography, the ancestor of bioregionalization using brachiopods. And this bioregionalization has really held a big grip on Devonian correlative studies, because it was such a huge step forward. And I don't want to take away from the work that was done there, because it really was an incredible step forward for we who study global correlations and biogeography, because these bioregionalizations gave us a suite of biotic and abiotic characteristics under a hierarchical label, which allows us as scientists and paleoscientists to have a shared taxonomy and a shared nomenclature. And so we could finally begin to talk to each other about these abiotic and biotic interactions and talk about them at different scale, but communicate across boundaries and know intuitively what was happening. So as you can see in the image, there are different levels, and provinces, put simply, are biogeographic units at a more local scale. And it takes multiple provinces to build a region. So that's level 1, and then an umbrella. And then multiple regions are then covered by the final umbrella of a realm. Some places do put in domains, but I will be primarily talking about realms, so I'll stop there. So when I started doing this, I really had to grapple with something that I had noticed when I was doing my Devonian studies. And that was that there was no real definition for the biogeographic areas within the Devonian. So Gondwana, as mentioned, was like 70% of the continental mass, and it moved between different latitudes and different environments, and there were obviously near shelf and far shelf, and yet everything, including their taxa and evolutionary milestones, they were incorporated into revisions that were based in absence. And what I mean by that is that instead of characterizing these huge and dynamic areas by what was present, they were characterized by what the old world had or didn't have. So for example, the Gondwana was always listed as less diverse than the old world realm. It was colder than the old world realm. It lacked important taxa like the brachiopod Tropidoleptus. And so, and I know that we can strike that last characteristic straight away because that's been, disproved pretty thoroughly. But I think that we should think about the underlying factor of each of these, which was that Gondwana was only ever characterized in comparison to and in absence. So it's not what Gondwana had, it was what Gondwana didn't have. And because of that, 70% of the land mass, of the continental mass at the time, was given one big blob of a definition, which was the Malvinocaphoric. And this is an enormous problem. So I think that this is also really helpful for in highlighting this, really shows the difference between a consensus and a diagnosis. We all agreed that the Malvinocaphoric was sufficient, and then we left it. And we used it to talk about our areas and the interrelationships between those areas. And it was useful, but it's not good enough anymore. And so I would go to conferences and I would meet people like Cameron and Renato, and I would have these great conversations, and we would complain. And then slowly things did begin to change. Look at this, isn't it beautiful? So meeting people from other globally south areas, I was able to see that the problems that I was seeing in my Australian data were being reiterated everywhere, from Brazil, you guys, and across in South Africa even. And I really hope I'm giving no spoilers, because Cameron will be talking later. But it was he, our South African colleague, along with D.A.T. Harper, that really formed what I would call Gondwana's first useful presence-based bioregionalization. And this was an extremely exciting moment. And it brought about the name change also of Malvinocaphoric being turned into the Malvinocrossen realm. And I think that we should all work really hard to make sure that we keep it as a Malvinocrossen now. So now that we have our foundations out of the way, we've seen that Gondwana, biogeography and taxonomy has really been influenced by Eurocentric definitions. And we get to see how absence-based definition stems in part from sampling and observer bias. But I'm also going to talk about modern examples. I'm going to talk about some modern examples to highlight the problem. And I'm going to use Australia because, I mean, we're pretty great. So this is my big problem, and I call it the bear in the room. Now, Australia has the oldest continuous culture, and in the language of the Eora Nation, this lovely little critter is called a koala. But when Europeans made it down, they saw it as the world's cutest and tiniest bear. And we can't really blame, these poor Europeans for their terrible identification, because they came down under with their Eurocentric identification booklets and dichotomous keys, and they had all the benefits and drawbacks of a European university education. And they made their identifications based on the knowledge they'd had and what they'd seen before, just as we all do. And they fitted this cute little koala into a slot within their understanding that worked best. And so to this day, some people call this a koala bear. Now, modern science, of course, knows that what this cutie really is, which is, of course, a marsupial. But when I was going through the trilobites of Australia and the bioregionalizations, and I kept seeing how the trilobite species and genera were like related to the old world realm. Time and time again, they were coming out closely related to Germany and Czech. And given that Australia at the time was on the other side of the planet, it didn't really sit right with me. So when we have a misidentification, if we were to call this a bear, then its migration history looks pretty similar, comes from Europe, spread across, came through Asia got slightly smaller, and then arrived in Australia. What a great narrative. I mean, of course, that's the dispersal history. It makes a lot of sense for bears. And this kind of thing is what's, was being told about Devonian trilobites too. But when you change up the taxonomy and systematics and you figure out it is actually a marsupial, and with more dedicated study by local scientists, Western scientists found out what the Eora Nation already did, that the koala is in fact a koala. It had a pouch and was much more related genetically, evolutionarily, and also geospatially to those things around it. Not to European bears, but to wombats and to kangaroos and to South America and Gondwanan areas, because that's its evolutionary history. And so this simple change in taxonomy really changed the area relationships of the koala and of Australia. And it sounds really simple when I say it like that, but weird stuff happens when you get to Australia, where the geographic and evolutionary principles of areas, where geographical proximity and them being the closest related to each other, it just doesn't function here. And I personally believe that that's because post-colonial areas are magic, and they can subvert the rules and accepted rules of evolution. Or something else is going on. So recognizing that a koala is a marsupial, it's not a bear, changing its geophysical relationships, we're able to reclaim Australia, reclaim the koala, and fit it back into something that actually functions, like what Penn, Clark, and Harper did. Beautiful stuff. So now that we've got that all sorted, we're going to talk about some of the underlying factors. So I really hope that I've convinced you that this problem with taxonomy and geographical bias founded in observer and sampling bias is a real problem, and that it's happening, and it's happening now. And I'm going to argue that in part it's because of colonialism and cultural imperialism. So colonialism is the policy and practice of acquiring full or partial political control over another country, occupying its settlers, and exploiting it economically. And cultural imperialism is the offense of an economically dominant culture on others. So what I'm saying, or trying to argue, is that if you've come externally to a new place to study it, you've brought the money, you've brought the ideas, you've brought your education, and you're studying it within your own frame of reference, which means if you're not from the local area, you're more likely to identify a bear than a koala. So sampling bias has been shown time and time again, and this was a study done by Raja et al., which quantified the amount of collections within a given country and how those collections are formed. So it's not just, oh, there's so many in America, there's so many in Europe, but actually how many were done with collaboration of domestic scientists, how many were exclusively external scientists coming in, and how many were exclusively local scientists doing it. So this is studied from the paleontology database, and I will be using the paleontology database for what follows. So bear this in mind as we go forward, because you can see that the wealthier the country, European settled, most of the time it's going to have a high amount of collection in the paleontology database. And the parachute index was another thing brought about by Raja et al. In this great paper, and it shows the amount of science done on a country with no local collaboration, no local scientists. So this really is one of those like cultural imperialism, you come in, you study, you take away kind of things. And so there are I'm going to guess that there probably are quite a few of Devonian bears. And these are some of the areas that are worst affected by it. And I'm sure you're beginning to notice that they are primarily in the global south. So now we get into proving all this. And I'll be sticking with the paleobiology database for a while and focusing, of course, on trilobites. So I just want to have a caveat that, of course, I love the paleobiology database. It's a great resource and has really opened up paleobiological research and created much more accessibility. But that said, we've seen that there are problems, and I'm going to highlight that there are probably more. So this is the spread of the paleo PBDB trilobite data for the Devonian. Pretty reasonable spread. We can see some lacking areas down in the global south. It's pretty poor around Siberia, but all in all, there's a pretty good spread. And it's quite dense in European and American areas. So what I did was I got all these Devonian species, and for every species that was sampled across more than one geopolitical boundary, I counted that as a co-occurrence. And I chose geopolitical boundaries rather than by geographical areas because, one, Gondwana doesn't have enough definition, the current bioregionalization requires revision, and two, it can be directly comparable to the work of Raja et al. 2022 with sampling and collection numbers and parachute index. And three, this way we are across the entirety of the Devonian rather than revising and changing bioregions when they were terminated. So to keep it simple, I found the co-occurrences, and for every co-occurrence, I marked whether they were related to proximal areas. So if it was a Gondwanan taxon, if it was this co-occurrence was found in Gondwana, then it was be counted as present. And then the second is if it was related to an external area. So it could be the global north, it could be Asia, depending on which side, say for Morocco or in Australia, then it's going to have a different count. And so the graph here shows the proportion of taxa that was shared with other Gondwanan areas or northern areas, given that they were co-occurring. So if a trilobite were found in two locations, the orange marked self shows the amount of taxa found within Gondwanan areas that cannot be found within northern areas, and vice versa for the north and Gondwana. Now that I've explained what this data means, I hope you can see a problem, which is that less than 20% of Gondwanan species do not co-occur with any other Gondwanan area. They co-occur exclusively with European and American areas. Compare that to what's happening in the north, where the majority of them co-occur with proximal areas in the global north. To me, this looks like a koala bear distribution. Not an awesome stuff. So species are hard, I understand that. So let's change it up the scale. my favorite thing to do. And we tend to use genus for biogeographic studies. Just covers a lot more of the sins of sampling bias and things. So, okay, well, we've had a look and we can see that the pattern is actually reiterated again. Yes, it's a little bit better. We're over 20% Gondwana to Gondwana relationship, but it's still nowhere near the kind of parity that you see in the global north. Indicative, just indicative, So let's compare species and genus. As expected, species way worse, genus way better. But this whole thing, like being able to define Gondwana by absence, suddenly seems really reasonable because there's nothing to define Gondwana with. All of the species, the majority of the species, relate directly back to Europe, directly back to these globally northern areas. So what could we possibly use to define Gondwana? We use the tools that we have for global correlations. But if my theory is correct, then part of the problem is not that Gondwana is empty and not that it needs an absence-based definition, but that we're misidentifying our taxa as bears and not as koalas. Our trilobites are related to the north when they should be related to us in the south. So I think I've shown that we have some really related problems and that koala bear distributions are feeding into these Eurocentric relationships when we should be getting koala distribution. And it's leading to these absence-based definitions, which we really don't want. So how did we get here? How did this idea of absence persist? I'm going to use an Australian example because I think it's really important that we look at the history and legacy of what's happening. So personally, with colonialism and cultural imperialism, there is legacy, and we've all been influenced by our teachers, and they've been influenced by their teachers. So if there is an academic lineage that is external to the region, for example, Australia with Britain, then there are natural influences on our natural sciences. And as we move forward, we really need to remember this idea of bear in the room. But I will be focusing on Caranelius and how the cultural legacy of the idea is still locked within Australian science and Devonian paleobiology. So Australia has the oldest continuous culture on the planet, and the Indigenous people were a really diverse group with over 250 language groups and over 800 dialects. But when Captain Cook arrived on the southeast of Australia, he declared terra nullius, which means simply that the land is empty and belongs to no one. So apart from being a bananas racist thing to say, this notion was really hung on to, was only corrected in the 1970s. So it's within literal living memory that Australia was empty and its Indigenous people were not people, but flora and fauna. And it's so sad that Australia is still seen as an empty landscape. Absence was all that was possible within Australia because Australia was fundamentally waiting to be filled. And so in early Australasian biogeographic studies, we can see already with the idea of the koalas migration through Europe to us, that this kind of thing is what was consistently happening even with the trilobites. Because when these kinds of ideas were being proposed, the world was static. They didn't have plate tectonics at that point. And so the ways that things was the way that things had always been. And so they're coming from a center and they're reaching out. And the things that reach out get smaller and smaller and smaller because ultimately the weakest goes to the wall. So primitive animals, smaller animals, more depauperate communities were found on the edge. And that's where Australia was seen to be, on the edge of the world. Same with many places in the global south. And so whilst this is obviously not a great idea and has been disproven time and time again, the idea has really been hung on to. And so we get these absence-based definitions and we get these koala bears. And I keep bringing it up because I keep thinking it's incredibly important but because it was ultimately just a justification of how European taxa were found and named again in Australia. And we don't just get that in modern taxa, but also in the Devonian. So there's a lot of work that needs to be done, whether it be structural or in terms of, foundational taxonomic changes. So I'm going to bring in now my own group to show how taxonomy can really change up some of these. So this is the Homonodidae. This is a really cute one. And it's pretty common down in Brazil, and I would love to come and look at them all, because I hope to create a phylogeny. I've done some pretty bad ones recently, so got to keep working. And they have a really nice shovel face. They have an excellent spread, excellent traits, and they're found throughout the Devonian. And so here's a rough estimation of their spread. Once again, I used geopolitical boundaries as why there's a bit of a dense cluster in Europe, but across the eastern seaboard of Australia, there's a few locations you can find them, the same for South America and North America. And when plotted, I have to say that the spread does look a lot more consistent, so it's okay. And more importantly for me, additionally important, was that they have a really good spread of facies. And when I'm considering as a biogeographer distribution, I'm really interested in how a clade reacts to abiotic events across a range of habitats. And it makes, to my mind, the signal that they give off in the data very, very robust. So I really wanted to compare how a taxon that I know very, very well, that has been revised exceptionally twice in recent history, how that compared against the work I did with the paleobiology database trilobite data. And what we can see is that the 1975 revision by Tomzakawa, I'm so sorry if I said her name wrong, in 1975, really does look like what we've seen in the paleobiology database, with quite a low relationship in Gondwana of self to self, and a much higher external relationship to more northern areas. And then it kind of evens out in the current revision, which it was done in 2005 by Sanford. And I think that this really highlights some of the positives and importance of taxonomic works and revisions, because by doing more work, we're actually beginning to uncover what's probably closer to a real pattern. And we're going to start being able to move away from definition by absence. Now, I would like to caveat this, of course, because a lot of the relationships here were Australia to Morocco for Gondwana to Gondwana, which isn't ideal, because they're also have a fair amount of cultural imperialism impacting their science and who's doing their science. But it's still an excellent start. So, put simply, the work that local scientists do on local areas is probably the antidote to discovering bears when we're looking for koalas, discovering Wendorfia, and finding it in places rather than, what used to be the extremely widespread Homonodis. And so we need to keep working on our local areas and keep driving down that parachute index and making sure that we're putting our hands up and making sure other people aren't taking our type specimens away from the country that they're found in. Now, it's easy enough to say that this is a problem. So I decided to do something similar to what Raj et al. Did and create an index, and I call it the KBI, the Koala Bias, or Bear, depending on how cute you feel, index. And it's a numerical estimate of taxonomic observer bias for paleontological data sets. And if you are interested in it, there are going to be two sets. And set one is using raw proportions, and set two is using Bayesian probabilities, using naive Bayes. And ideally, they will be weighted for, as you will see, they will be weighted by area. So we'll start here with the raw proportions KBI, and it'll be used to highlight the uneven sampling and to strengthen intra rather than inter-area relationships. And if you worry about it, here's what it looks like, which is the proportion of taxa shared in area and interest divided by the proportion of taxa shared in total times by 100. And this can be weighted by area. So if you were to weight it first, you would expect Gondwana to be 70% of the available, then you could times it by 0.3, something like that. If you're worried about what that looks like, well, you don't need to because you've already seen it. It's what I've used time and time again in this talk, and it just gives like a really simple way to communicate the likelihood of a koala bear in your data set. So now that we've seen set 1, and I think we have a pretty good grip on it, we move into set 2, which is Bayesian, and I think this is probably. I think that the raw proportions are probably more powerful, one, because they're easier for people to use, but also easier for people to understand. But the Bayesian will give us probabilities, which I think is important for more, if you're looking to predict or suggest significance of a relationship as well. So the question for the Bayesian probability is that will a taxon co-occur in contiguous areas as opposed to non-contiguous areas, given that the taxon co-occurs? Don't worry too much about this. We've probably all seen it. Quite a few of us have like statistics fear, but I'll quickly show you what that would look like. So it would look like the proportion of taxa that co-occur in Gondwana times by the proportion that co-occur over co-occurrence in Gondwana and co-occurring plus the northern and Gondwana relationships times by the proportion of northern. So it's just a way of like drawing in all the different vectors, and this can be done by a series of percentages and dot points, and it's fine. So the data around the co-occurrences, we get a single number, which is why it's probably nicer for some studies than set 1 of the KBI. And for our current data set, we only get 1.9% probability of a co-occurrence of a Gondwanan taxon in a Gondwanan area. 1.9%. That's pretty bad. That's not good. So hopefully either set 1 or set 2 could be a really useful tool for databases and global correlations, and to really give some caveat to the correlations that we are seeing, give us language and metric to throw into the ring when we see a global study that underrepresents 70% of the Devonian continental mass, which is of course Gondwana. But do bear in mind, this currently is a draft. The paper and metric is in the works, and I'm working really hard to try and get people from the paleobiology database involved so that it can be like chucked straight into the data and people can look at it and start questioning historicity and legacy and how the data set itself is flawed. So I'm almost out of time. I think I've used up some of the time for questions as well, but I think that I've also brought a lot of problems, and so I'm just going to quickly go through some suggestions. So we really need to reduce parachute science, and that means putting our hands up, making sure that we see people who are working on our local areas and making sure they don't work on those local areas in our time without us. We have valuable things to comment, and that's probably one of the most powerful, simple ways that we can advocate for the global south. And then, of course, if you're working in bioregionalization, please, please, please define by presence. And if you see any global studies that underrepresent Gondwanan areas, publish a comment on that. Like, call that stuff out. We can't be sitting by anymore with this absence-based definition of Gondwanan areas. And positionality just means that if you are concerned about your interpretation, and of course just being like, my education is X and I'm coming at it from this perspective. Something that they do in what's that science where they study people? History. Yeah. But of course, these are individual solutions to a systemic problem, and we have to start somewhere, but we need more. And so for some broad scale solutions that we really should be aiming for and lobbying for is free translation into English of scientific works and back from English into your local language. Like, we are providing our science to publishers for free. I think that we can ask for them to translate our words so that they can be interpreted by as many people as possible. Open access fee waivers, look them up. If you're from a country with a low GDP, then you're more likely to get access to these waivers. And then you can, bring your science out and have a much more wide, everybody can read it in your home country as well, which is going to be great. Reduce the movement of type specimens out of country of origin. And when I give you a proper reference, please use the koala index for large data sets so that we can really begin to question how we do global correlations. So thank you very much. Koalas are really cute, but they're pretty unhelpful if you're doing global correlations. And I hope that I've given you some things to talk about, but not too much. And I would love to thank once again you all, and especially the organizers. I cannot wait to see all the talks. And welcome again to everybody to the first Gondwana Devonian Symposium. It's fantastic. Bye. I'll be here for questions.
Hi, Elizabeth.
Hello.
Fantastic. Thank you very much. Thank you very much for the talk.
Hi, Elizabeth.
Hi.
Hello, everyone. Hello.
Well, nice talk, Elizabeth.
Hi.
Hi.
Hi, Cameron.
I don't know. Too many microphones.
Yes.
There are too many
Please,
microphones on my class.
1 minute, please.
Okay.
Elizabeth, thank you so much for the talk. It's a pleasure. Found.
Yes.
Yeah.
If there are.
And
Yes.
we have time to one or two questions.
I would love to hear them.
All right. Mercedes.
Where do I find the questions?
Elizabeth. What do you think?
Well, I also appreciate very much your presentation, and it's open to questions. I don't know
Yes.
if Renato has.
Yes, I have a question, Elizabeth. What do you think about well, you talk about this, but the language is a barrier in your country. The database of trilobites, principally, mainly in Homalonothides. It's a group. It's a group that we have 2 or 3 species here in Brazil, but some problems, some troubles about the taphonomic preservation. This preservation interfere in your database. What do you think about this?
I think it's incredibly likely that in the actual raw data, of course, there will be taphonomic biases towards certain areas and environments. And that was why I was really trying to show some of the spread of facies, because then we at least get some idea of what kind of facies they're likely to be preserved in, and we have some buffer, I guess. But dealing with taphonomic biases and preservation biases is something that you are going to have to deal with in literally every single kind of data that we'll be working with as paleontologists, because we simply do not have the same kind of spread or consistency of data that you would have in neontology, for example. So yes, I completely agree with you. And you can do heaps of ad hoc analyses, but personally, I like to look at the data for what it is and look purely at the pattern that I see within it. rather than try and do some fancy mathematics to smooth out these lines and form more of a gradient, because even in modern ecologies, they're not as often a smooth gradient between communities. Sometimes it can be more hard. So I'd rather just deal with what the data is right now, rather than do some ad hoc or analyses on it to make sure it's smooth. I hope that answered your question.
All right. Any questions?
Yeah, I have. Oh, sorry.
Please.
Oh, now that Cameron also raised his hand, so
Cameron.
You can go first, Gilda.
Okay, thank you. So my question is if you have already any feedback how the scientific community is dealing with your koala index. And by the way, the name is really funny. I really love the analogy. Basically it's that it. If you already have any data on that. Thanks.
So I'm actually currently working with a person who studies Paleopleistocene bats, and they also have very similar spread of data, where when you actually track the changes in species distribution with revision. So I think that this was probably like the most important thing that I wanted to show, is that the more that you study an individual taxon, the more that the distribution of the taxon changes. That has nothing to do with where the species are found, but it's this biogeographic dispersal narrative that we put behind our data. And I think that having that definition, like the difference between, you know, what a consensus is and what a diagnosis is, and how that relates back to the data that we're looking at, the evolutionary history of the species that we're looking at. And so, yeah, there are people who are working on bats that I've been speaking to, and I've also been speaking to people who work on diatoms, and those can be both modern and paleontological, I guess, because I have a colleague who does all of that. And so we're just trying to see whether the best way to represent the data for this publication that I'm looking to put it together would be to compare between the biogeographic spread of the different revisions, or to compare that to what we see in the paleobiology database, or if we can create some kind of code that would automate the revision dates, and then see how, if there's a more koala-based distribution with older revision dates versus more new revision dates, so then it becomes a bit more predictive, per se. In the works.
Cameron.
Hi. This is an excellent presentation, Elizabeth. How's it? And I hope all is well on your end.
Thank you.
From what I can tell from your presentation is, it's something that I've been thinking about as well for some time, is that we actually do, as Gondwanans as a whole, need to look probably a full revamp of the taxonomy of virtually anything from Gondwana. So itself, it would be a very big task. It would probably require several lifetimes worth of work. I think it would be worth it. And also would require a lot of cross-continental collaboration, so that we actually work in different collections and different material, especially to pick out any of these nuances that we could have, is that anything, you know, could be intraspecific variation versus interspecific variation, etc., etc.
Yeah.
And I think this would be a good thing, is that to break down any, especially these very large imperially imposed legacy barriers such as colonialism that we all are dealing with, the ghosts of the past. We really are. We can't escape it, but we should fix these things. Mm-hmm. So, do you think maybe it's a question to pose to the entire community? Maybe it's a bit too early to bring it up now, but do you think as a whole we should maybe push for one of these, UNESCO-funded IUGS type projects to at least begin funding sessions such as these in future to reunite Gondwana as such, to bring us all together in the same room to talk about these ideas, and then also as well perhaps even maybe fund new research? Do you think, in your opinion, do you think that this is something that we should go for as a community? And
I mean, of course, that would be fantastic. You know, it would be fantastic to get as many of us in a room and to really find out ways to, I guess, target key areas and bring that together. But I definitely think that I can be as enthusiastic as I want to be, and I think I'm a pretty enthusiastic person, so I'm always up for a new challenge. But that's definitely going to have to be a bigger question, probably for everybody here. But if I were to be a person who was like beginning a grant proposal, I'd probably be like using things like the KBI or the parachute index. We could tie these all together to show taxa or areas that really require the most funding and require the most assistance in taxonomic revision. So I think that if we're starting like beyond the idea that this might happen, then I feel we already have sufficient metrics and ideas to kind of highlight the necessity of the center. Great idea, very fun.
All right, thank you very much, Elizabeth. Again, it's a pleasure.
No worries.
Thank you very much. Well, we need to go away, and the next presentation is by Mercedes.
Hello. Now it's time. Then I continue, Renato.
Sorry, Mercedes.
So, are you going to? Você vai colocar a minha apresentação?
Yes,
or?
yes, your presentation is here.
See? Okay. Então, I'm going to present.
Just a minute.
Yes. This work that we started with my co-authors in 2014. Now it's published in the chapter of the book Brazilian Paleofloras that is more or less finished, although some chapters are still under editing. So this is one of these chapters, still not completely available in the final editing form. And well, the presentation I have here.
Do you see the presentation, Mercedes?
My presentation. Do you want to? I try. Let's see.
Mercedes, just a minute. Where is the presentation? You see the presentation on YouTube?
No, I am in YouTube. Okay.
It's better for you?
The problem is that I don't see very well, but
all right. Não,
now
vou retirar a apresentação, Mercedes.
No.
Você quer passar por aí?
Não, eu vou olhando pelo WhatsApp.
Não há problema. Não há problema.
Yes.
Perfect.
No problem. So well, I will present the palynologic analysis of the Barreirinha formation, Famenian, bearing Proto-Salvinia from southern margin of the Amazonas basin, Brazil. And as I said, in collaboration with William Matsumura, who is present in the room, and also Roberto Iannuzzi. And our presentation, if you can go next, please. So you have to help me to Renato para pasar.
Sorry, Mercedes, there is a delay.
Yes, I will say next. See?
Right.
Okay. Well, then thank you. No. As an introduction, perhaps it's not well seen in YouTube, but it's small. But Proto-Salvinia was described as an enigmatic plant-like organism of possible terrestrial origin. The genus includes 5 species that are documented in the Famenian, the late Famenian, not latest. One is Proto-Salvinia brasiliensis and Bailobata. from the Amazonas basin. And 3 others are Ravenna, Furcata, and Arnoldi, that are known from the Kentucky, Michigan, Illinois, and Appalachian basins of North America, and also in northern Brazil. According to Niklas and Phillips, 1977, Protosalvinia ravenna and Furcata are forms that can be ontogenetically derived from P. arnoldi stage. Whereas other authors, in this case Niklas and collaborators, 1977-76 as well, consider all forms of Protosalvinia as ontogenetic stages and ecotypes of only one single natural species, Protosalvinia brasiliensis. Niklas and Phillips, they say that, they suggest that the observed pattern that we show in this slide of form distribution of Protosalvinia is the result of paleoecological factors along the marginal to central parts of the basin as a function of depth gradient, and a similar pattern is registered in assemblages of northern Brazil and also the North American basins. So it seems that occurs in the 2 regions. So the next slide will present in the next slide. Okay, thank you. The results of the palynological analysis that we did in the lower shale section of the upper part of the Uraria member of the Barreirinha formation. And the samples were collected from an outcrop along the southern margin of the Igarapé Azul, São Florencio, Florenico, corresponding to the locality 12 of Niklas and collaborators, collected in a channel of the Rio Tapajós of the Amazonas basin. This section is characterized by interbedded and interlaminated siltstones and shales bearing Protosalvinia, which is also morphologically reanalyzed terrain due to its importance as a fossil guide for the Famea in North Brazil and USA. Also, I must tell that Lobosiak et al., collaborators in 1997, published a single palynological study, the only one, of the interval bearing Protosalvinia from boreholes like the Caima PH2 of the Amazonas basin. In the next slide, we are showing in the next slide the piece of rock selected for this analysis bearing Protosalvinia, mostly in the upper surface, as it is shown. This rock consists of 3 laminae, 2 gray laminated shales sampled for palynology, so the upper and the lower, and in the middle there is a fine sandstone with current structures, which was barren for palynology. So in the upper shale, numerous compressions of Protosalvinia preserved parallel to the stratification plane. Some small compressions of cuticles of the Protosalvinia were detached using steel needles and a stereo microscope, and were cleaned using concentrated HF, and also remains were obtained from supervised maceration with this HF in a petri dish under the hood. Some complete specimens of Protosalvinia and cuticles mounted in stubs, non-metallized, using low vacuum, were illustrated in a Phenom Pro SEM. Two samples for palynology, which are marked in red circles, were processed with standard methods and slice mounted with Shelly glycerine and also with water to take fluorescence and SEM pictures later. Well, in the next slide, I am showing the morphology of the results obtained of the morphology of our specimens, more or less ranging from 300 to 500 microns, the ones selected and pictured. And they confirm the multicellular surface pattern of dense variably rounded to polygonal cells we see in these pictures. And also the uni to multilocular reproductive structures and bearing the samples, the palynological samples, yielded also megaspores that are illustrated in the next slide, that were described by Phillips et al. From the Famenian of Kentucky and also for the Amazon basin. And we see here then this pattern of cells, and to the right the Protosalvinia megaspores, which are over 200 microns. These megaspores are thick-walled, and they are called Retusotriletes sp by Lobosiak and collaborators, and also Rooney et al. Defined as Retusotriletes lobosiaki, being the same species, no? The arrow, which is white in the specimen below, is marking an Umbellapheridum saaricum, an acritarch, which is embedded or mounted into the megaspore. It's not well seen, of course, but believe me, is there. We must say that several works were done to try to identify the botanical affinity of this plant, and especially Quijada and collaborators in 2016 has made an analysis with several techniques, and the biomarkers finally reveal its vascular plant affinity. Not completely precise, but being a vascular plant and not algae or brown algae of the group Phyta, as other authors previously reported, no? So in the next slide, I am showing the palynologic results that were provided in this analysis as well, and especially to the two samples. One, in the lower shale, yielded abundant amorphous organic matter and less frequent cuticles of Protosalvinia and other phytoclasts, as well as palynomorphs represented mostly by phytoplankton and also spores. The other sample, the upper surface with a lot of Protosalvinia, presents a higher proportion of phytoclast derived from this Protosalvinia, a less proportion of amorphous organic matter, and fairly well preserved palynomorphs represented by phytoplankton and spores. A total of 40 so in the next, we see in the plate here several acritarchs, and those marked in red are really diagnostic, age diagnostic. So in this another two plates with spores that are also showing diagnostic species marked in red again, and especially several are also documented in the latest Famennian assemblages bearing Retispora lepidophyta, which is not present or not identified, not found in these samples. So in the next slide, I will say that next slide, that the biostratigraphic range of the species in the next slide, the next slide.
2 minutes, Mercedes.
See. Not that one, a previous one. I think it's a... But it's more or less the same, I can say, yes. So you can read, the ones that can read, that the biostratigraphic range of these species, especially those that are diagnostic of this shorter range in the Famennian, allowed us to establish a correlation with the mid-late Famennian Rugospora radiata and Bayatis porites hystricosus interval zones of Melo and Lobosiak, defined in Brazil. Of the 77 species determined, 25 species are shared exclusively with the Bayatis porites hystricosus interval zone. So with the confirmation of the absence of Retispora lepidophyta, then our correlation is with this zone, no? Bayatis porites hystricosus, particularly. So now see the next slide with the chart of biostratigraphic correlation, show us that the floral stage Protosalvinia, defined just in the presentation in the poster of Matsumura, that we are sharing with you through the Google Drive folder. And we ask everybody has posters, sharing the poster there in the Google Drive, so we all be able to look at them. Well, this floral stage corresponds to the or it's equivalent to the Protosalvinia zone defined by or described originally by Lobosiak and collaborators in 1997, and also to the Bayatis porites hystricosus, as I said, interval zone of Melo and Lobosiak. And this correlation of Brazil into Brazil is also extended to the European plant zones of Cyclostigma and Brachyphyton, defined by Edwards, and also the open zone called Verrucosa istricosus of the Ardennes regions by Strill and collaborators, as you see there. Instead, in USA, the Proto-Salvinia zone is a little bit larger in time and varies from the middle to late, not latest, again, not with Retispora lepidophyta, Famenian, Diducites versabilis, Grandispora cornuta, and also the other one I mentioned, Verrucosa and Istricosus palinozones, which are correlated to the conodon zones, Marginifera and Lower Expansa. Well, it's spanning around 3 million years. So in the next and final slide, final, I want to say two things. One related to the paleoenvironment of the position of this assemblage, which correspond to the shade interval, devoid of dimetites of the Uraria member. It is interpreted for us as deposited in a brackish environment under euxinic conditions as a result of increasing freshwater influx, supported by the mixture of marine phytoplankton, organic amorphous, and pyrite, and also the terrestrial palinomorphs, cuticles, and tracheids found associated. So this Proto-Salvinia could be interpreted as a living plant into these brackish coastal swamps, but also could have been transported to the depot center. And well, finally, and concerning the paleogeographic distribution of Proto-Salvinia, as we see in the paleogeographic map, this is the only plant element recorded in the late Famenian of the Amazon and Parnaíba basins of Brazil, and also in Michigan, Illinois, and Appalachian basins of North America, as I said before, possibly representing a single floral stage due to its high abundance, limited stratigraphic range, and wide regional distribution, although closer regions, of course, associated with also the trace fossil Zoophycus, and bearing also similar palynological composition, along with the presence of the megaspore Retusotriletes lobosiaki. So, well, one explanation for this is that the narrowing of the Rake Ocean since the early Devonian times triggered the exchange in this moment of the late Famenian, very much closer the two northern and southern hemispheres, reinforcing a worldwide distribution of the late and latest Famenian palynomorphs, and also Proto-Salvinia, and later, not far from, the appearance of Retispora lepidophyta, also confirming this widespread of fossils between northern and southern hemispheres. Well, this is, I'm sorry if I extended more than the time I have.
Thank you, Mercedes. Nice. All right, Mercedes.
Yes, thank you.
Okay, thank you. Unfortunately, we don't have time to questions, so
So I think you have to continue with the other two presentations,
yes,
right? Okay.
yes.
And I think one is Shilda, no? But first,
Right. Presentation is John Marshall.
mhm.
Yeah, presented to us here in Brazil.
Okay, perfect. I will deactivate the microphone.
Okay, so I guess online you probably won't see me, and I can't see you. Just rearranging some slides. Yeah, that's the one. So, just as a quick bit of background to this, then, so I'm covering for John today, who I believe is sat online. I'm his PhD student. Is that working? Okay. All right. So I'll start again. So I am covering for John Marshall today, who is sat online. I'm his former PhD student, and I started with him 20 years ago, working in Bolivia, and we'll get to that. And the other author here is John Lakin. He was our PhD student, and he was working the Lake Devonian, also in Bolivia. So these days, I'm a little bit lost to the Paleozoic. I live and work in Rio these days, which is why I'm here, because I was in the neighborhood. I spend my days working on the Cretaceous, which I consider topsoil. I'd much rather be doing this. So we're really grateful for the opportunities, particularly me, because I'm avoiding lots of meetings today to be here talking to you, which is much more valuable and interesting to me. So thank you very much for the invitation to come and present today. So next slide. So we're going to cover the work that was really started by John in the late 90s, mid to late 90s in Bolivia. And then, as I said, carried on through myself and John Lakin. And I've sort of come back to it recently. And so really all I'm going to describe here is just the basic problem. I think that Elizabeth touched on it. I think some of the key messages we get out of this presentation in respect to what we're doing about sampling bias, geographic bias, here in South America. And the big problem is that the mission of the Devonian Subcommission really is to put the Devonian section onto an A4 page, it's shown on the right here. And these days it's about increasing the definition of and resolution of the stage boundaries. It's about identifying sea level changes, relative sea level changes, geochemistry cycles of evolution and extinction. That's good. That's great. The problem is, though, is a lot of the stage boundaries and the data behind them, they're defined on groups that are very rare in South America. So the goniatites and the conodonts do exist, but they're quite rare and also poorly constrained. So that's the problem. Next slide, please. So down here, we were sitting at high paleo latitudes in the Devonian. It's tectonically pretty quiet. We have a series of intercontinental seaways in South America. We don't have any climatically sensitive sediments like carbonates. In the absence of carbonates, in part, one of the problems that we have with the goniatites and the conodonts. And we largely have an endemic fauna for most of the Devonian with quite unique fossils, generally isolated. Next slide, please. So let's hop over to Bolivia, the western side of South America. So as I said, John started work here in the mid to late 90s, mainly on samples. And then later on, I came along to look at the surface exposure. So the thing about Bolivia is that you've got the exposure because of the Andes, thrust tectonic setting, everything gets stacked up, holding in the faulting because of the subduction of the Nazca Plate in South America. and you have sections that are kilometres long. The Devonian is highlighted here, and in the areas where I was working, it's about 2.5 kilometres thick. It's important because one of the major hydrocarbon reservoirs in the area was there. That's the Wamam Pampa Formation. And there's also some source intervals in the Los Monos Formation and the Formation, as shown on the right here above it. So that's how things stack up. The problem that you've got here, as I said, is tectonic. So the picture on the left, 2 photos, this is Abra del Condor in southern Bolivia. So it's mountainside, it's hillside, it's very pretty, but geologically it's quite complex. So there's a sort of a very simple interpretation of what's happening in that section underneath. You've got 2 major thrusts, you can fold in and minor thrusting in between. So when you've got that level of complexity, and you've got thick plastic shales, sandstones, how do you really know where you are? Which complicates everything. So one of the things that is working quite well in South America generally is palynology. And what John noted in his earliest work was that there was when you go down the Devonian section, you encounter a consistent succession of quite distinct fossils and palynology. And the spacing of those polymorphs and where they appear, those structures, is quite well spaced at the moment. We weren't sure at the time what the environmental significance of them was, though, which is why we went to the surface to look at them. So the section I worked on, or started work on 20 years ago, was a section called Bermejo, which is sat just a few hours west of Santa Cruz in central southern Bolivia. Now, I mentioned the events that you can see that the consistent palynomorphs, the distinctive palynomorphs, that 4 of them are shown here. Youngest to oldest, left to right, Petrovina crenata, Lucidia canarense, Vimergia bansoni, and Ivitia someri, which will be the case study for this talk. And on the panel at the very bottom there, you can see how they stack up, youngest at the top, oldest at the bottom, tracking from north to south across central southern Bolivia, so about 100 kilometers. You've got that consistent pattern of those events. The yellow interval there is what we call the phytoclast dominated interval. That's something else. If we get time, we can talk about it. But what I'm showing on the top there is the expression of the Ivitia someri event exposure at Bermejo. So at Bermejo, we go from the Wamam Pampa Formation, which is the sandstone reservoir I mentioned, and then you've got the Los Monos Formation sitting on top of it. This is a marine transgression. You're going from sandstone to offshore shales. And in the middle of that transgression, you see Ivitia someri arriving. It dominates the assemblage. Everything else disappears, and the slides, the samples that you take through that interval, are dominated by Ivitia someri. And so the graphs on the left there, top left, you're looking on the left-hand side, you've got a section of about 40 meters. And you can see the numbers of someri, they're flatlining for a few tens of meters, then you get a big spike, and then it's gone again. So this is quite a distinctive event that we see at exposure, and again, it's replicated in the subsurface across Bolivia and northern Argentina, and now we've got a context for it. We know that it's associated with the marine transgression. Important in the context of the Devonian timescale on an A4 slide earlier on, because we can start to think about relative sea level changes in Bolivia and how they tie into the global sea level curve that's been defined previously. So, next slide. So, we have the someri event. I mentioned as well that goniatites are rare in South America. They do occur though. There was a published record from southern Bolivia, Campo Redondo, shown as a strat section from Campo Redondo in the middle there. Hunican et al. In 1980 found goniatite, which was assigned to an Upper Juvetian Franian age range. I went back to that section in 2004 and I logged it and I did the palynology on it, and I also looked at the macropaleontology in that section, and that's the stick of rock on the right hand side. It runs from the early Devonian Ichla Formation up into the Wamam-Pampa sandstone reservoir, and you've got a bit of Los Monos sat on top of it. I also found a goniatite in that section, bottom left there, it's shown. Now the important thing about that, in contrast to what's come previously by Hoonican, is that this goniatite is actually carbonate. And goniatite workers like to break those open, and they prefer them to be preserved in carbonate because they can see more and they can classify them better. The Hoonican specimens were molds, decalcified, much more difficult to work with. So we gave this goniatite to Thomas Becker, and he quite definitively told us that this thing cannot be any older than the early Eiffelian, but it certainly isn't an Upper Givettian Franian goniatite. It's much older. It's a homeomorph of something that comes later. So, context of Ivitia someri then. We know that someri is associated with the marine transgression, and we started to track it around Bolivia, and we found it at the Campo Redondo section, and also we can tie Ivitia someri into the Chacahiri section, which is close to Sucre, so heading towards the southwest of Bolivia. And also in that section it occurs in the Carinatus shale, where the brachiopod Tropidolectus has been identified as well. So we're building up a picture now, and we're integrating data from palynology, well, from geology, lithostrat, we're going through the macro paleontology, and we're doing the palynology, and we're linking everything together now. So it's across Bolivia. Next slide. So the marine transgression I've covered already, but I will say a little bit more in that the type description of the Ivitia someri comes from northeastern Brazil in the Parnaíba Basin in the well 2PM1MA. And there, you probably can't make this out so well, but in the map there is the well section from that well, and you're going, the transition from the Itaim to the Pimentera Formation is sandstones to stones, so transgression, and you've got someri sat in the lower part of that marine transgression. So demonstratively, you've got the same assemblage that we see with Bolivia. It's the same thing. So that transgression you can take across South America now. So it's looking quite widespread. But in the bigger picture, we also have other distinctive palynomorphs with a more cosmopolitan distribution that are found in Euramerica and northern Gondwana that we also identify here in Gondwana. And this allows us to tie and put the Devonian sections in Bolivia into bigger context and be a bit more confident about how we're splitting up those sections, and also be a bit more confident about whether or not we're seeing truly global sea level change and events here in South America. Here are some of the other palynomorphs that we see. We won't go into detail in those, so we'll go to the next slide. So, with what we've got in Bolivia and how we think things tie together outside of South America, John has also worked in other places, Grand Malvina and South Africa. And with some of the key palynomorphs that we see in Bolivia and also some of the macro fossils that we see, we can start to tie these other areas in as well. So I quickly touched on Tropidolectus in the Chacahiri type section for the Wamam-Pampa. We use Tropidolectus as well to tie into other areas like South Africa. And based on where we've got the constraint for it with the someri event in Bolivia, and we've got the age dating through other palynomorphs and also the goniatites that we found. So when we go back to the Devonian events and the sea level curve, what we think we're doing, we're sort of tracking the Kajak event of Euramerica across South America and into South Africa, based on what we're seeing. So that's the sort of early mid-Devonian story that we've got going here. Next slide. But it doesn't sort of stop there. Based on the lithological trends that we're seeing and our correlations with the biostrat, we can also identify other events later in the Devonian. So this is John Lakin's field area up near Lake Titicaca in northern Bolivia. And where John is stood there, he's on a sandstone. I don't remember the formation names up there. I think this is the Cumaná and Casa, where he stood on a glacially striated pavement and a diamictite to the right-hand side of it. And John Lakin did a high-resolution study, geochemistry and biostrat study across that interval, looking for the DC boundary. And in summary, what we've got there on the left-hand side is a 30-meter log section with all the sample points across approximately where John is stood. And what we see there is a quite diverse cosmopolitan Devonian spore assemblage, Rugospora and Retispora lepidophyta, grapnel tip spores, and acritarchs kicking around. It's quite diverse. And then all of a sudden, our TOC drops off. We get very few spores, plankton in the assemblages recovered. Things blank out. And then with increasing stratigraphic height, there's a bit of a recovery. We start to see low diversity, palanomorph assemblage coming back in, spores and prasinophytes mainly. And what we think we're looking at here, we're going across the association. You've got the extinction, things disappear, and then they start to come back in again. So it's well constrained. We can't exactly put it's this point, this is the exact boundary, but we can go to within a few 5 or 10 meters of where we think the boundary should be. And I think that's probably good enough. So that's the other aspect to what we do in Bolivia. We've covered kilometers of section. We've got thousands of samples that we've looked at. You know, still sampling bias, and we've integrated data across scales. We do recognise that there are other complications in play that do affect our interpretation, like tectonic. We have to be careful with stratigraphic repetition. It is very likely paleo latitude and indeed paleoclimate to some degree are causing local complications in the sequence. But when you have a big data set, you can think about these things carefully and be data driven in your interpretations. But the good news is at least I think within the Gondwana basins, there are some events that are unmistakable that we see. So some of the marine transgressions and also things like the DC boundary, they're quite clear. So that's a good thing. So next slide. I'm coming to the end here. So what we've done then is over the last 30 years or so, we've developed a high resolution biostratigraphic data set for the Devonian of Bolivia, pretty much covering all of it. We've also gone down into the Silurian as well more recently. And we've done our best to integrate our surface observations with subsurface data. We've linked the geology back to the palynology and the paleontology. And I think we can clearly demonstrate the applicability of some of the Devonian events as defined at lower paleo latitudes in the sea level curve in South America and also beyond. And you might not be able to see it here at least, but we also took this work outside of South America. We try to identify the South American palynology in other places. We worked in Morocco, we've looked in the UK, we sampled sections in Russia. Gilda will talk about Spain in the next talk, and that's a slide from I don't know, 15 years ago when John and I were looking to identify Voltisfridium sommeri of Kramer in that area. I guess Gilda will say more about that. And also more recently, during the lockdown at least, here in Brazil. So naturally it follows. I'm going to go and look at the Devonian here as well for fun. Go next slide. So during the lockdown here, and perhaps pushing home office to the limit because we weren't in the office. So technically, I could work from anywhere. I actually dropped into the Paraná Basin a few times to have a look at the sections at Jaguaribe and also in the Tibagi area. Next to me in that photo is Kawan Juk, who works together with me at Equinor. He's a bit of a geological geek like me, and so he took me out and he's from Curitiba, so he knows the area, so he took me out to have a look. And while we were there on this section on the PR 340, colleagues from Unicinos were actually in the area and came over to have a conversation with us. Matthias Vargas and Zephyr, and they were there, also describing sections. So, I think that's a good thing. We're here today because we work on the section. We want to share our work with you. And this, I like this photo because this is collaboration. I wasn't expecting to bump into anybody in the field, but colleagues were there. It was a good conversation, and really that's how it should be. So thanks again for inviting us, and we could talk for a good fair few more hours about this. So if there's any invitations to other events or universities, I'm more than happy to get away from Cretaceous and come and talk to you all. So thanks a lot.
Fortunately, we don't have time to questions. I have some questions to you, but another time. Well, we let's continue with the. Oh, sorry, I don't see.
With Shilda Lopez.
Okay.
Shilda Lopez, she will present the next talk.
Yeah, and I would like to ask for a favor. My image feed here across the Atlantic is taking more than 10 seconds to change.
Okay.
It has a quite big delay. Can I share my screen?
Okay.
Okay, are you guys watching it? Are you guys seeing the presentation?
No, I see your face, Julia.
Okay, that's strange. Let me try it like this. And what about now? Is it okay?
No, Gilda.
No?
No, see you again.
Okay, so it's better if you guys just present it and we'll go from there. And I will manage, don't worry. Okay. So now I will have to wait a little bit. I'm not seeing anything in the YouTube channel. Okay, but I will go even if I'm blind, so you can let me know if I'm moving too fast or too slow. So I'm going to present a project that we are currently developing on the Devonian of Spain. In this first slide, you can see quite a lot of names. These are all the people that are working in this new project. So
Gilda,
next slide.
Just a minute. I don't.
Yeah.
We need that our presentation is here in Brazil or.
Yeah, it's better in Brazil because if you are just seeing my face, no one will get anything from that.
Sorry, just a minute.
No, that's okay. You will just have to bear with me when the slides are transitioning because it will be a little longer between slides for me to speak.
Just a minute.
Yeah, thank you.
Judah, you see?
No, but like I said, I will go blind and you can just follow me through the presentation. Okay, but are you guys seeing now?
All right.
Okay, so let's start again. Okay, so I'm going, like I was saying, I'm going to present you the new project that we are working on the Devonian of northern Spain. So there goes another plus for colonialism and our bias. So you can see quite a lot of names in this presentation. These are all our group elements that are working in this project. Next slide, please. And that I'm going to talk a little bit more now about all our roles. So basically, our project is to work with the polymorph assemblages from the American terrain in Spain. Between other things, we want to have accurate and updated polyclimatical and polyenvironmental reconstructions of this area of the globe during the Devonian. So in this project, there are actively 6 people working on it. So we have 3 palynologists. Our PI at University of Sheffield is Charlie Wellman. The other palynology is John Marshall, and I've joined this project this February, also as a palynologist, more focused on the marine, so in the aqueducts and chitinozoans, while Charlie and John are mostly focused on the spores and cryptospores, even if John is actually helping me also with the aqueducts. We also have Jason Hilton from Birmingham, is our paleobotanist, so he's helping us to establish correlations with the macroflora from this region. We are also focusing on anoxic and extinction events, and I will be talking a little bit more about that in the following slides. So for that, we have David Bond, that is our geochemist in this project, and the person she's the last to be presented, but not the least, because she's basically gathering all the data from all of us and putting into nice models so we can do nice paleoclimatic and paleoenvironmental reconstructions of all the data that we have for this area. Is Sarah Green from Birmingham also. So we have several questions. In the following slide, please. We have quite a lot of questions that we want to solve, but 2 of the main issues that we want to actually focus more is related. The first one with the palynological assemblages. So basically, the American terrain was a set of islands, so it was an archipelago that we believe had an intermediate position between Euro-America and Gondwana. There are quite a lot of schools that have different views on whether the American terrain was more located closer to America, and others believe that it's closer to northern Gondwana. So after studying all the polynomorph assemblages, we are hoping to actually provide some answers that could help us locate this set of islands in with a more accurate position. So that's one of the focuses on our project. Also, like Ian just talked about in the previous presentation, conodonts and goniatites are actually generally absent from South America. So one way to establish correlations within Gondwana, Euro-America, and that also includes South America, obviously, is precisely using the polynomorphs. And we want to use not only the continental as well as the marine polynomorphs that we are studying from this section. So that is one of the main focus focuses of our project. So now, talking a little bit of why we chose this area to sample and to study. So here you have the location on one of the paleogeographical models, the location of the ATA, so the American terrain. We have a very nice set of islands where actually the Southwest Britain and America were also located. However, these two areas are quite tectonically deformed, so that can be that is actually an issue for our material. And from studies that have been published in the 60s and 70s, and I will go through that briefly, we know that the Cantabrian Mountains can actually or have quite a lot of interesting material and well-preserved material to study. We could also go, obviously, to the northern part of Gondwana, to Morocco. We have Patagoniatites and conodons, but that, in terms of correlations, for example, with South America, will be a flop. We also know that the structural deformation is also less compared to the other regions in the American terrain. But when we are doing studies on palynology, and with this picture you have a very nice image of the sections. They are all very well exposed. However, they are deeply weathered, so to work with it's a nightmare. And also throughout the geological time we had a hotspot under North Africa, so that can be a problem because we have everything just cooked. Next slide, please. So, like I said, there has been quite a lot of studies done in the 60s and the 70s by Fritz Kramer and Carmine Kramer, his wife. So we know that what type of material that exists there, or we have an idea. But this references also have quite a lot of pitfalls, and I'm going through some of those in the next slides. So we know that there were quite an interesting material. We have new species, quite a lot of new species described from these works, and not only continental material, as well as marine material. And as a curiosity, I can say that it was in these works, in some of these works, that the first cryptospores were actually described from this region in Spain. They were not called cryptospores back then, but this is where we can find the first descriptions from this region in Spain. So, the next slide, please. This is the area that we are sampling and we are working with. It can be quite complicated in terms of tectonic assessment. However, in the regions that we are focusing, we still have quite thick carbonate sequences with well-characterized conodont faunas from our Spanish colleagues. And we can actually establish a study where we can have a marine shelf transect, so we can have a good spatial coverage of our assemblages, as well as in terms of age, so vertically. We are focusing more on the Asturia-Leonese zone, so we will be working with, in particularly with those successions. Here you have an idea in this map with the lithostratigraphic units of the successions that I'm currently studying. So I'm more focused now in these months in the lower Devonian and in the La Vide group from Lyon area. That includes a succession that ranges from the middle Oakhoff to almost the top of the Enzian. So another important issue that we want to approach in our project is also related to extinction events, like I told you about. So basically, we want to use several power-environmental proxies and also stable isotopes to try to understand how the marine and the terrestrial polynomorph groups responded to these extinction events in this part of the globe. So that is something that we are really looking forward to. So these are some of the sections that we are currently sampling. For example, here you have a really nice one, a carbonate sequence from the coastal areas. Next slide, please. And here you have a photograph of a section inland. They are quite accessible for sampling, and you can actually see in the picture, I hope, that we have a road there, so they are quite easily, we can quite easily sample the sections, even inland. Next slide, please. So here you have a picture of the Lavid group type section near the Lavid village. It was one of the sections that were already sampled and that I'm actually currently studying. In this section, we can sample is represented almost all the Lavid group. So it's one of the most complete sections that we have in our study, and is well correlated in terms of age, can be correlated with the Conodonts. From the works of the Kramers, we know that the Lavid group is also one of the most productive, especially the Valpor-Carroll Formation, so in the middle to upper Andean. But as I told you before, we cannot currently fully rely on the works of Kramers because they are not updated, taking into account all the knowledge that we know currently. Also, unfortunately, like I said, quite a lot of new species in general were described in these works. However, the slides with the specimens are lost, so we cannot study them again, even if we want to. Also, from what we can the information that we can retrieve from those works indicate us that they probably did quite a lot of spot sampling, so that's not the best when we are trying to do a detailed biostratigraphic work. And the ranges were never properly defined when he was characterizing the genera and the species and describing them. So that are some of the pitfalls on its works that we are now trying to update and actually do a proper biostrat work, among other studies. And another detail that I wanted for you to take a look is that red arrow that also indicates us, like you saw in the geological map. It is a quite complex tectonic zone. And in this case, in this photo, we have an inverted section also. So Charlie and Ian and John, like Ian just referred, have been working there for quite a long time. There are several sections that were already sampled. We are doing more sampling next month, so we will be going to Spain. And the assemblages from the slides that have already been studied show that both in marine forms as well as in the continental groups, we have quite a lot of data to study. And in the successions, we have a good preservation and highly variable thermal maturity. And what do we know? So some of the sections I've been studying by Charlie and John for quite a long time. So in this case, I'm going to show you just very briefly because we don't have time. The data on the continental material, so on the spores that have been already been published and studied. So with the spores, they were applied coefficients of similarities and Jacquard indexes to see the variation in terms of paleophytogeographical arrangement. And we can see that overall, there are much more similarities in terms of assemblages, spore assemblages with Gondwana than actually Euro-America. And that may indicate us that actually the archipelago is much closer to Gondwana. Does that happen also with the marine material? Well, that's something that we are looking forward to figure it out in the next years. Next slide, please. So these are some of the material that has been already studied. So in this case, the spores, we have very, as you can see in the plates here, quite a lot of different spores. Next slide, please. The thermal maturity can be quite variable. However, still quite good preservation. Next slide, please. Here again, simple spores, Azambitis sporites, for example. Next slide. Here we have not only the Infanis sporites that are quite beautiful, as well as examples of cryptospores as Artemopyra. Next slide, please. And here we also have monolith spores at the bottom of the plate, as well as more cryptospores that are represented here as Cnidospora divelomedia. Next slide. And what about South America? Because this symposium is on South America also. Well, with our work, we are planning to find key markers that will be able to be correlated not only with South America, as well as with other regions within Gondwana and Euro-America. So, like Ian just talked about, key markers as Evitia someri and Bimurga can be some of the markers that we want to find in our material. We know that Evitia was reported by Kramer in one of his works in this area. However, we have never seen any more examples of this species so far. So let's hope that in the future we can recover some of these specimens in our samples. Next slide, please. And obviously, with all the data, we are hoping to establish biozonal schemes or event schemes as detailed as these ones, for example, that compare Europe with the Amazonas basin. In this case, we have the continental biozonation from Melo and Lobosiak and Pleferendino. So let's hope that our data will be enough to do such a detailed correlation and actually be a point in the globe that can correlate several areas or several countries within the Devonian. And next slide, please. And that's it. Thank you. I don't know if I was rushing too much or not, but I think you got a glimpse of the presentation, and you can just ask the questions later on, both in Portuguese or in English. Thank you.
Gilda, muito obrigado. Infelizmente não temos tempo, mas eu agradeço muito a sua presença aqui. Thank you so much.
Obrigada, Gabriel.
Vamos agora à palestra do final dessa manhã do Cameron. Eu peço, por favor, se a Mercedes estiver aí para fazer a introdução, por favor.
Sim. Bom, it's a pleasure for us to introduce Dr. Cameron Penn Clark, who is going to present the lecture. Are all events rooted by same cause? event stratigraphy in the Devonian of West Gondwana and the pitfalls of correlating this with global tempos. He focused particularly in Paleozoic brachiopods, and currently he is a honorary research associate at the Evolutionary Studies Institute in the University of Witwatersrand in South Africa. So please, you can start. Thank you.
Good morning, everyone, from across the Atlantic. It is a great honor to have been invited here today as a guest speaker at the first of hopefully many more Gondwana Devonian Symposia. Today I will be speaking about the event stratigraphy of West Gondwana and the merits and pitfalls of correlating these events with global tempos. I hope you enjoy. Just a quick disclaimer before we begin. This presentation and its contents are the sole property of the Council for Geoscience. The opinions expressed herein may not be representative of the organization and as such are exempt of liability. So how this presentation is structured today will firstly be a broad overview of the Devonian period, zooming in on observations from West Gondwana and their interpretations and linkages with global events. And lastly, their merits and pitfalls and how we might be able to resolve them. So as we all know, the Devonian period, two major supercontinents, Laurasia and Siberia, with a small little continent towards the north, and Gondwana towards the south. Throughout the Devonian period, both... I think I'll just jump on in. I think there's a problem with audio. Sorry about that, guys. So I'll just skip ahead over here. This is just a global reconstruction of the Devonian. And yeah, it's showing convergence taking place there between Gondwana and Laurasia. Associated with the formation of Pangea. Gondwana drifted northwards with closure of the Rex Sea very early on in the Devonian, and followed by almost like scissor style zipping up of the Paleoclitocene towards the late Devonian period. What we do see also in terms of temperatures is we see from what we can tell, is that climates were more or less equitable, similar to what we've got today, where there were distinct latitudinal belts ranging from warm temperate climates at the equator or near the equator, northwards into more arid tropical type climates, and then towards the south and probably the North Pole as well, more cool, temperate and cold climates. What we can see with the temperature record in the lower right-hand corner is that temperatures in the earliest Devonian were quite warm, becoming increasingly cooler, moving into the middle Devonian, only to increase again into the Fronian and then dropping down towards the Flaminian. Normally, and this is probably associated with the late Paleozoic glaciation of Gondwana in the Carboniferous. When we look at sea level trends from the Devonian, the Devonian period, we can see at different sort of scales of what's going on. So what we do see is that sea levels were quite high. So this is just to zoom in over here, just to show you what the Devonian period seas look like. So what we can see is that sea levels are quite high, but at the same time, we see a trend of decreasing sea levels from the Silurian into the Carboniferous. This is again probably associated with the formation of the Carboniferous late Paleozoic ice earth conditions. If we have to zoom in then on what is normally taken as the reference sea level curve for the Devonian period, this is from more low latitude, this is from North America, so this is a Eurasian type sea level curve or Laurasian. What we can see with the actual trend is of decreasing sea levels during the earliest part, increasing sea levels in the middle Devonian, only to decrease again towards the latest Flaminian. So what we can see in the observed trend is rising sea levels within a net first order fall in sea levels. Again, this was also a time period of, there's a lot going on biologically. So what we do see is plants make their sort of first real rooted foray onto land, starting with these very simple plants, diversifying to the first sort of forests, true forests, by the end of the Devonian period. And this was followed by the concomitant invasion onto land by arthropods. In the oceans as well, we see the appearance of ammonoid cephalopods. Fish undergo major radiation, so we've got the first appearance of true sharks, ray fin fish, as well as low fin fish. These were coexisting alongside older sort of orders of fish, so placoderms, acanthodians, and jawless fish. These guys went extinct towards the end of the Devonian period. And if we look at lobe-fin fish, is that a lineage of lobe-fin fish gave rise to tetrapods towards the end of the Devonian period, which also made their first brave steps onto land. So a lot is going on biologically during this time period. What we do also see as well is that it was also a time of great sort of global level endemism. We see is that, especially in the oceans, is that biogeographically life seemed to coexist within distinct regions. So the major regions that we know about are the Eastern America, Norvino-Capric, and the Old World realm. These seem to have been around from the early to middle Devonian, probably collapsing somewhere around the Javetian or Iphilian Javetian, with the trend of more cosmopolitan type faunas as the global condition going into the late Devonian, as well as being joined by the recently recognized West Gondwana bioregion. With all this biological activity going on, there's also changes in the rise of new environments and ecosystems. So in the oceans, we see this widespread development of biogenically mediated reefs. Corals chromatoporoid reefs, they extend to their greatest ordinal extent, or into their aerial extent, great diversity. So you can see that you get this changeover from more archaeocyath, or these archaeocyath reefs, they went extinct during the Cambrian, of course. Quite a distinct changeover. What we also see as well is a change in the way in which animals fed, a changeover in the marine realm from things being more substrate type feeders towards things that fed in the water column, so the nectonic revolution. So a lot more free swimming animals that populated the water column itself. So I guess with all this, there were new niches and new opportunities for animals to exploit different niches. So this again led to changes in food webs, nutrient cycling, and it's essentially what we see today as the modern condition, having its genesis in the Devonian period. On land as well, things also kicked it up a bit. So we see it's a rise of terrestrially adapted plants, embryophytes, what we know of today on land, alongside other smaller plants, biophytes, etc. And what we do see is that these plants had a founder effect, encouraging animals to move on to land. This has also started the beginnings, the genesis of terrestrial-based ecosystems and food webs that we currently exploit, as well as other tetrapods and other arthropods, etc. And this new biological activity created new pathways for weathering, and the development of the first true soils also led to the development of new terrestrial geomorphic terrains, so switch over from more braid plain type fluvial environments to meandering rivers, and also played an important role in the sequestration of mud to marine basins. So what these figures are showing you is this, of course, this changeover in fluvial style. But importantly, if you see the graph over there by McMahon and Davies, you can see that there's a lot more mud being recorded on terrestrial platforms. So it's again, with all of this, there are starts of new biogeochemical transfers between life and Earth. We also see it's also a time of great extinction, for lack of a better word. So quite a few punctuated extinction events. There are about a dozen of them. They're not really well understood. The major sort of impetus is always put on the late Devonian extinctions because they took out the most biomass, I guess. So what we do see is with all of these extinction events in the middle and lower Devonian is that they usually are quite short term. In the marine realm, anything that is benthonic was severely impacted. You see a large spike in plankton and nectin. We also see as well that there seems to be a tie-in with changes in sea level, which also seem to have brought in dysoxia, and there are significant geochemical anomalies. So what is also interesting as well is that with these extinction events seem to be associated with major biotic events. So I guess the greening of land, but also as well with probably with Milankovitch cycles as well, or with these two working in symphony with each other. So yeah, the jury's still out with a lot of them, and these could also be extraterrestrial. So we don't really know a lot about these extinction events, but we'll get onto that in a bit later. So when we also think about the Devonian period, what you probably can gather from this, and what I've gathered from this, is that most of our current understanding is from former equatorial and low latitude regions. And there's quite a disproportionate amount of information from polar regions themselves. So this is just taken from PaleoDB. This is all known fossil occurrences from the Devonian period. And as you can see is that northern hemisphere regions, which would be Laurussia, sort of lights up like a Christmas tree, whereas the former Gondwanan regions, excepting parts of South America, South Africa, and Australia, are quite sparsely populated with known data points. This is quite disconcerting in that there's a large, landmass. West Gondwana itself is situated around the South Pole, so there's actually a large chunk of the global picture that's completely missing. So this is just a reconstruction map to show what these regions, or what the South or Paleo South Pole looked like during the Devonian period, and we can see is that for most of the part of it was flooded over. This large, very shallow Peric seaway, with several depot centers dotted around West Gondwana, the majority of them being on the South American side, as well as North Africa, to a lesser extent, southernmost South Africa, the Falkland Islands or Malvinas, and West Antarctica. Also as well, when we think of high latitude regions, we also have a skewed mindset as well, is that generally the first thing that jumps to mind is that we always think of high latitude regions, the polar regions, as being iced over. I guess we are living in an ice house time period, and not a greenhouse time period. So just to flick back over here, you can see is that you don't see any ice caps over the Paleo South Pole. So we also know as well is that some variability exists across these latitudinal belts in the present day, but we don't sort of ask, was this the same in the past? And we know as well that events that happen at we know in the present day is that the events that take place at the South Pole, the present day, where both the poles have a very strong influence over seasonal bio-events and probably major bio-events in future and bio-crises. So are we able to compare the past, the present with the past? So this is what our talk is about today. So what we can see is that if we have to delve back in time and look at time slices, is that West Gondwana was ice-free for most of the period. So these are just time slices from the Silurian Devonian boundary, moving through to the Praguean Juvetian times, and then into the latest Devonian, the Flaminian, where one starts seeing the development of polar ice caps just before the late Paleozoic ice age. And so we do see scant evidence from Bolivia, Brazil, and from South Africa as well. So the jury is still out, as well as that there might have been sea ice at the Paleo North Pole. So recent climatic modeling studies that have been done by Ulrich and Butzka and Burger seem to suggest that there could have been a mass of sea ice at the Paleo North Pole that could have been regulating temperature. I'm not too sure that if there's any other evidence for it. I mean, I would expect to see grounded ice like we do today, but this could be for a bunch of other factors, which we'll get through in a moment. So what characterizes all these West Gondwanan depot centers? So they all are chiefly siliciclastic in nature. They are all pretty much storm and wave dominated depot systems. So these shallow siliciclastic seaways, which were probably anywhere between about 50 to 80 meters deep. So they weren't terribly deep, very, very shallow, went on for days. And what we also see is that, whilst the rest of the world were celebrating carbonate reef development, we didn't have that luxury. So this is just a reconstruction of what the Borkenfeld Sea would have looked like, which would be the equivalent of the Parana, and looked like quite a peaceful place. What we also see as well is that it's sort of when we have the development of these seaways in the Devonian, it seems as if it happened more or less at the same time. So what we do see is that there's this first order subsidence and transgression across West Gondwana, some probably starting somewhere in the Praguean towards the Jubetian. In South Africa, it seems to linger a little bit longer into the late Devonian. All of these basins are intracrotonic and episutural in nature, so we're not seeing any evidence for divergent drifting type motion, plate motions at all. These things seem to be, for lack of a better word, these thermal sag, or I don't want to use that word too liberally, but these sag type basins, so intraplate type basins. There's also a very remarkable similarity in the stratigraphy across these depot centers. So this is from a recent paper from Vargas and colleagues from Brazil. And what we are seeing over here is that there seems to be a match in depot styles across all of these basins. So when we look at the sequence stratigraphy, we again see a very similarity in stacking patterns and in basinal shifts. So it does seem to point, if our calibration is correct, and again we'll get to that in a moment, it does seem as if there is a shared possible driver, and there seems to be one. The Thamatinian orogeny seems to be more or less the right age for this massive transgression to take place. So when there's this renewed activity taking place, as the pre-Cordilleran phase, it seems to be coincident with this large transgression. What we also see as well is that other than the similarity in depositional style and in depositional environments, we also see that there's a similarity in marine diversity trends. So this can be broken down into three very loosely defined biodiversity epochs. There's this Locovian fauna, which sort of sits over there. There's this Praguean Emsian fauna, as well as this post-Eiffelian fauna. What is interesting to see as well is that where these faunules, or these biodiversity epochs are located, they're located in sort of downturns of global temperature. So wherever, the graph itself shows a decrease in temperature, it seems as if this is when these faunas tended to be around, only to disappear with an upward tick in temperatures. So there does also seem to be this sort of correlation that one sees in West Gondwana. So just to summarize these different faunas, the Locovian faunas, they seem to be largely these Silurian holdovers. So there's this, it's only in a few places in West Gondwana that one sees this, so the pre-Cordillera, in Guinea and Senegal, so in the Bovee Basin, as well as possibly Bolivia and maybe even in South Africa. This would require us to just verify. But what we do see is that there's this eclectic fauna. It's a mixture of Afro-South American endemics, as well as North Silurian endemics and new novel forms, so mainly Kentronites and Samonites. And this is just a strat column from Herrera, from the pre-Cordillera, in the Talacosta Formation, and one can see this quite nicely, this sort of division. There's this clear Locovian fauna separated, separated by the Malvina-Clausen fauna, which comes in at the Emzian. By the Praguean faunas doesn't seem to be much in the way, but again, this is if we've got our dates right, which we'll get to in a bit. Then going to the Praguean, Emzian, Trifelian, what we do see in West Gondwana as well is that we see there's more of a regional type explosion of life, I guess, of these endemic regions. Traditionally split into three regions: the Old World, Eastern Americas, and Malvino-Catholic, and these are split into where they seem to coincide with latitudinal belts. And with the Malvino-Catholic realm in particular, this seems to be coincident with this transgression in the late early Devonian. This was recently redefined. So there were a few problems that we found with this. The first of which being that the name itself is of offensive nature, but also as well that there are better ways in which to describe the ways in which these fauna congregated. So we seem to recognize, myself and my colleague Dave Harper, that there are two large bioregions. There's this temperate latitude bioregion and this high latitude bioregion. And this was the Colombian, West African, the Amazonian, and the Malvina-Clausen bioregion. And essentially is, if you could imagine, the Malvina-Clausen and the Colombian, West African bioregion are quite distinct. This is in terms of their brachiopods fauna, but the Amazonian is like a true, it's a mixing zone, a true intermediary between the two, between these two end-member bioregions. So, and what we do see as well is that these true Malvina-Clausen endemics, they tend to be more of them towards the pole, or the paleopole regions. So, since I think most of us actually work on these quizzes, I thought I'll take some time to explain. We'll talk about them a little bit more. I really enjoy brachiopods, so I can go on forever. But anywho's, just in short, the Malvina-Clausen brachiopod biota, you've got strophomenates and rhynchonellates. There are several of these. These are just the endemics. But in short, it appears as if certain of these could have, these organisms, or these brachiopods, have got some relation to similar Eastern American faunules. Some of these animals as well, so mainly Anabia harringtonia or Harringtonia, seem to be the ancestor of ancestral form of Ostrocelia. At the same time, Anocena seems to be ancestral to Samonites and Kentronites, which sort of persisted into Malvina-Clausen times. This can also be expanded as well to include several endemic trilobites. So among thacopod trilobites, colmonids are completely wholly endemic to the Malvina-Clausen bioregion. There are several delmanatids, homolognatids, and protoid trilobites. These seem to be genus and species specific to the region. Ostracods, not really sure. As of yet, not much work has been done on them. Bivalves and gastropods, these seem to be more or less cosmopolitan, but I could be mistaken. There are one or two genera that seem to be unique to the Malvina-Clausen bioregion. And then echinoderms themselves, they seem to be a whole other kettle of fish, but there's a great similarity with regions elsewhere, and endemism seems to be focused on species-specific taxa. But what we all know is a commonality is certain things are quite abundant. So conularids, especially here in South Africa, conularids are quite common. High lithids, especially in the lower parts, I guess. I mean, that's what we're seeing over here. They seem to be quite common. And then tentaculatids are virtually everywhere. Corals and bryozoans are very rare, but there are some things that are odd that we just don't have. So granule-type ammonoids, yes, we do get the odd one or two critters that probably, wandered into the basin, floated in. So granulotypes are, I would like to say, rare to absent. Graptolites, we don't have any of those, and no conodonts. So these are major things that the rest of the world uses for biostratigraphic constraint. We don't have them, which makes our lives difficult and interesting at the same time. What we do see as well with the niches that these animals lived is that they more or less seem to have liked colder type waters, so cool to colder type waters. There's a strong modality towards benthic lifestyles. You do get, one or two, the only nectonic organisms that we see other than fish, I guess, are also ceratid nautiloids. And there's a strong association to live in these shallow marine silicic environments, mainly in these subtidal regions below wave base. Seems to have been the prime property in West Gondwana during the Devonian. Then what is actually very interesting, and it's just when I look at a lot of the data that keeps them coming out of Brazil, is that just how similar things are across the Atlantic. So what we do see is that there seems to be, when we look at these organisms through time, is that there's this massive sort of they seem to reach their diversity quite early on, so during the Eocene, and then show a gradual decline in abundance moving through into the Arthelian and into the Javetian. So what is interesting is that in both Brazil and I guess other regions in South America as well as in South Africa, is that what we are seeing is that these changes in biota, their abundance and the number of representative genera seems to be correlated with changes in sea level. There seems to be virtually no rates of regeneration rates seem to be null, so it seems to be a trend of pure decimation. We're also seeing as well this diminutization of biota, especially when you get to the terminal Arthelian. There's also this trend when the Malvina-Clausen collapses that there's this apparent appearance of Tropidoleptus, but pretty sure I put an asterisk there. There are problems with that statement, which we'll get to in a bit. But what's interesting as well is that the ecological response that we are seeing from the trace fossil record is that it does seem as if this was a real thing. Is that there are even changes in ichno-assemblages. What that means, I guess we're getting closer to finding out, but it seems as if these guys were quite stressed. All of this seems to be working in tandem in the background with changes in from what we've seen over the years, that if you look at the global temperature record, it does seem as if the collapse of the Malvina Closum bioregion as well as surrounding bioregions seem to be strongly affected by changes in global temperature, as well as sea level. So we do see that there's this entrenched sort of level. The Slockovian Juvetian seems to be a sweet spot, this cooler time. So could there have been a circumpolar current that could have been protecting, I guess, sheltering these guys, sort of working almost as like an allopatric barrier. But we're also seeing, so that's just, where you have this decline and then this changeover. But what we are also seeing as well is that when we see especially these big changes in biotic diversity, is that they seem to be at first glance coincident with these what seem to be at first very localized extinction events in the northern hemisphere. They seem to be correlative with events now, or the events in the southern hemisphere seem to be correlative. So these two show strong correlation with changes in sea level. So what is going on over here? We don't really know. So we're moving into the last, these post-Arthelian biotas. So what we do see is the sort of trend of what I always call the lights on but no one's at home, is that when you look at the succession, for all intents and purposes, sedimentologically, you can't see any difference between this part of the succession and the lowermost parts of the succession, in South Africa especially, where everything seems to be the same, but there's just no life or very little life. So what we do see is that when you do find fossils, which is very, very rare, they seem to be dominated by Tropidoleptus. And then in the lower parts, these very small, diminutive-sized and very impoverished Malvina Closum fauna. The only real newcomer that one sees is Rhipidothyris. So these guys as a whole seem to completely disappear before what we think could be the Franian Comenian. Depending on who you believe what the range of Tropidoleptus is. But it's very intriguing to see similarity of trends in both regions or across the Atlantic. So this now gets into part 3 of the talk, is ask the question, are all these events rooted in the same cause? So is the same thing driving the same, or is it all happening at the same time, and is it caused by the same factor? So just to surmise, we see similarities in depositional style, sequence stratigraphy, and biotic diversity trends. These do seem to be correlative with global bioevents, global eustatic trends, global climatic regimes. But our big problem is, and this is a real big problem, in West Gondwana, we are constrained by we have a very, very poor chronostratigraphic control. So are these actually correlative? Let's investigate. So the first problem we have is chronostratigraphic constraints. There's nothing known as of yet that has helped define actual sim depositional dates. I've been hoping for years now is that someone would find, a nice, or several nice ash-cliff horizons, or maybe even a couple glauconite rich sandstones, for instance, to try and provide a nice sim depositional, if not very early diagenetic age for these successions, but alas, nothing. What we do see though is that we can get maximum sedimentation dates from the youngest retrieval zircons, and these do seem to show close approximations for what we infer the depositional ages to be. So again, what we're seeing, this is just showing a graph. This is comparing zircon populations from La Parana and Uruguay to the Ventana in Argentina, as well as the North Patagonian Massif and the Cape Supergroup of South Africa. And what these peaks are showing, you can see is that there's a similarity in peaks. So the major take-home message over here with this is that the detritus that has been shed into these basins is more or less the same. So rocks of the same age. So we've seen similar in provenance areas. And this is just to show as well, this is just recent, this is current work that I'm doing for my collaborator at University of Johannesburg. And what we have done, we're looking at these zircon populations quite intensively. And what we are seeing over here now, this is from the Borgefeld group, is that the youngest age fraction that we've got, so it's on the left-hand side of that distribution graph, seems to show this Devonian population that's coming in. So, and this is more or less with the bracketed age of what we think the Borgefeld group is in age. So around about 400 to about 390 million years of age. So just a bit of caution is that any sort of work that one does, I advise you back with some isotopic work to help zero in on provenance areas to also account for recycling and inheritance. Of course, remember these zircons have been recycled over and over again through successive erosive and depositional regimes, so just be careful. Problem number 2, biostratigraphic constraints. So the endemic nature of these biota, from what we can see, is right from these Locovian faunas through to the Malvina-Clausen faunas and even the post-Malvina-Clausen faunas, is that they are quite endemic. So this makes comparison with regions elsewhere very, very difficult. The taxonomy of many of these groups are quite shaky. I have seen that there's been this renaissance happening in South America, mainly focused in Brazil. Really well done, guys. You guys keep it up. Of this trend towards revisiting the taxonomy of these groups and revising their taxonomy, which is great. It needs to be done. So yeah, then going on to our friend Tropidoleptis. So in my honest opinion, it's a terrible biostratigraphic marker to use for the Devetian. This is a very sort of outdated idea, is that Tropidoleptis only makes its appearance in the Malvina-Clausen region during around the Devetian. What we do know is it is a long-lived taxon, and it is actually quite ubiquitous and a very opportunistic taxon. So what we do see is that it seems to thrive when it is the only brachiopod around. So in South Africa, we find Tropidoleptis right from the base of the succession, right from the Gerdo Formation, all the way up into the basal Wittenberg group. It is just important to note, though, that it is not in great abundances when with the Malvina-Clausen fauna. It's only once these guys disappear that they really have no competition. So it's open field day for these guys. So then going on to the chitinozone galenomorph data, there's in recent years there have been quite major successes in extracting these fossils from these successions, both in South America as well as from the Malvinas. But they are quite difficult to extract. They're often very poorly preserved and also seem to be quite low diversity. I'm not a galenomorph or chitinozone expert, but from what I've heard from workers is that it does seem that even these little critters seem to show some level of endemism as well. So it's also a little bit of I almost wonder if it's a matter of the tail wagging the dog or the dog wagging the tail. Is that are we trying to match chitin polymorph data to what we think the macrofossil assemblages are in age, or vice versa. I'm not too certain of that, but it would be great though if we could get more of these from different regions. Then problem 3, looking at global trends during the Devonian. To call these global might not entirely be correct. So these trends are largely biased towards lower latitude observations. And also where does one fit in these curves? It's very easy to set up a transgressive regressive cycle, and you could put yourself in anywhere. So yeah, where do we fit in these curves? And also as well, what is actually driving things like we used to see at this time period? Where are the ice caps? We're in a convergent supercontinental regime. We should expect to see a drop in sea level, not this rise that one sees in the middle sort of Devonian period. And this extends as well also to other things like temperature trends, for instance. So cyclicity and observed trends, especially if we're seeing West Gondwana, are most likely probably due to local tectonics, the Famotinian being the probable drivers introduced earlier. And yeah, temperature trends, when we look at global temperature trends as well, again, these tend to be from data from low latitudes, from conodont appetite. I guess we can't point fingers since we don't have any conodonts to give these higher latitude estimations out. So this is also a bit of a bugaboo that needs to be solved. Then onto the last part of this last problem is what I'd like to introduce here are high latitude effects. So again, introducing as well is that a lot of the stuff we do know about, especially how the southern or the South Pole functions, is from glacial type epochs. So the present day glacial regime, the late Paleozoic glacial regime, but we don't really know in non-glacial epochs what's going on. So it seems to be quite underexplored. We do know is that these are quite sensitive environments, so they seem to be strongly affected. There's a combination of factors. So continental configuration, which we see today, is definitely a very strong factor in the environments and ecosystems around Antarctica. Orbital forcing, etc. These all will regulate things like ocean currents, temperature, salinity, etc. So how much of this can we take when looking at Devonian or at the Devonian period, especially with what we are seeing in West Gondwana? So I'll just diverge a bit just to explain what goes on at high latitude. So this is essentially, this is where the global food webs begin. So we do see in the world's oceans that there is a definite asymmetric production in productivity. So in that the majority of the biomass that one has globally is housed at the poles, and that your production is seasonal, and it changes with changes in oceanic density differences, salinity differences that again are a function of seasonality. So what happens is that, of course, we all know, many of us who live on the west coast of a continent knows about when you have these seasonal upwelling times as well, where all this nutrient-rich water is brought towards the equator side as well. So we do see these turnover events which help regulate ocean food webs, sets up the entire sort of positive feedback loop. In terms of climate, we also see as well as that there's of course a difference in solar irradiance. Seems to be, it is quite high at the poles, quite low, quite high at the equators, quite low at the poles, and this creates temperature gradients. So in the present day we see this. So as a rule of thumb, this is, I guess, why it's warmer towards the equator and cooler towards the poles. The knock-on effects of this, the Earth has to sweat, for lack of a better word, is that the Earth needs to transfer this heat somehow. So this latitudinal difference in temperature helps generate temperature gradients, which also will generate differences in just helps wind to blow. It also regulates latitudinal bands as well, where one has arid climates and humid climates. So the whole shooting match takes place because of this, the way in which Earth loses its heat or transfers its heat across its surface. Then we factor in so geostrophic flow being at its greatest at the poles and negligible at the equator. This is, of course, factors in with why things like tropical cyclones don't cross over the equator, for instance. But what it means as well, geostrophic flow, is that any sort of currents that are located around the polar regions tend to flow parallel to these pressure gradients themselves. So again, this drives oceanic circulation, and of course, oceanic circulation, this factors into ocean currents themselves, how ocean currents flow, how they transfer warm water and cold water across the globe, where they're situated, etc., as well as density currents. So what we do see as well is that in terms of salinity, polar regions tend to be more saline as opposed to lower latitude regions, and collectively this helps drive the Great Oceanic Conveyor, this thermohaline circulation of deep waters being brought up towards the surface, again spreading or helping drive global food webs. We look then again at currents and storms in more detail. You can essentially explain these in terms of low latitudes and high latitudes. So low latitudes, one tends to get tropical cyclones. These are driven by latent heat, so they're not frontal in nature. In the mid latitudes, one sees these mid latitude cyclones. They are frontal. Those are driven by definite thermal gradients as cold air as cold air chases. It literally chases like the warmer air. And then at high latitudes, for the southern hemisphere only, is that we do see these circumpolar storms and currents. They, of course, are frontal in nature, but these do appear to be a factor of continental configuration. So if the Drake Passage weren't open, would these actually exist? So this is again where continental configuration comes into play, not just temperature differences, but continental configuration influences these storms and currents as well. So at the end of the day, the knock-on effects of this is that you've got differences in precipitation and weathering. So polewards, you've got more mechanical weathering type processes, low latitudes, more chemical, humid, and biological mediated weathering processes, and this generates different clays, for instance, that are fed into marine, or eventually can feed into marine basins, but there are differences. And then probably last, and what's most important, I guess, is that in polar regimes that we see in the present is that ice sheets strongly influence global sea levels. But in non-polar regimes, the controls are most likely or purely tectonic and climatic that control global eustatic shifts. But then if you look at the tidal range of the poles as well, these are regions of net microtidal regimes. So the lower than average sea level implies a greater or possible susceptibility to changes in base level. So I guess if there was any change, or if there are any changes in global sea level trends, the poles would be affected, probably, would be affected, would be quite affected quite severely. So to sum this all up, is I've been giving this a lot of thought, and in order to try to flesh out and to try to solve all these problems, I do think we need to go back and systematically work through this entire story from base to top again. So we can start this by improving taxonomy. So what we should actually start doing now is we should start being more, we should start looking at if we do these sort of studies, is to be more regional in our approach, to actually go and visit collections in other countries, in other depot basins, etc. From there, we can probably read out any possible or probable intraspecific variation. I've got a feeling that the herd could be thinned out quite a bit, so we could actually see what true diversity is, what one sees, or we could see if any of these basins have got anything that's truly unique to them. I'm proposing as well that we look at doing more intensive and extensive palynological studies. Some areas will be tougher than others. South Africa in particular seems to be particularly difficult to try to get anything out of these rocks over here, but we are working on it. This is probably going to be the best shot we have at trying to constrain any local relative chronostratigraphy, safety in numbers, I guess. The more we find, the better. We should also then, once this is sorted out, look at improving intrabasinal biostrat studies. So it would be interesting to see, since we are seeing these trends, is that how similar are these really? Are the same taxa disappearing at the same time under the same set of circumstances? This would be very interesting to look at doing. And then more basin-wide sedimentological and zircon studies. So most depot centers around West Gondwana could do with thorough reappraisals. Very detailed work is required on them. And then going back as well, especially on this last point I made about continental configuration influencing currents and productivity, is that we need to also look at working on more up-to-date high latitude-centric paleogeographic studies. So we need to see if there's any potential intrinsic factors that were unique to the Paleo South Pole, especially given our continental configuration at the time, and that there weren't any known ice caps for most of the Devonian period. So what I believe you guys on, this has more been a thought exercise than anything else, where it's more of outlining problems than actually giving or providing solutions. On that note, I'd just like to say a very big bit of gratitude to my employer, the Council for Geoscience, as well as my affiliates at Wits University and my funders for assisting in this research over here, in particular the stuff I've presented from South Africa. So thank you. A big vote of thanks goes to you guys who've organized this. This is fantastic. I think we need to do this more often. Maybe we should host the second Gondwana Devonian Symposium somewhere else in Gondwana, maybe here, maybe Australia, or maybe even the Falklands, who knows. But I think this is good. It's great to get us to exchange ideas, and this is how we're going to have to work together to solve all these issues. So thank you guys for coming up with this.
Cameron?
I'm here.
Oh, yeah. Terrific, man. Thank you so